Hello everyone. Today I am going to discuss the topic of oxidative phosphorylation, the phenomena which occurs in the mitochondrial inner membrane. Suppose we already know that mitochondria is a double membrane structure. Suppose these are the two membranes of mitochondria. This is the outer membrane and this what I have drawn is the lipid bilayer. And suppose this is the inner membrane, this is again the lipid bilayer. So there will be an intermembrane space present between the two membranes always. Obviously, this part becomes the mitochondrial matrix. This is the mitochondrial matrix, and this outer part is the cytoplasmic part. Right? This inner membrane of mitochondria has a lot of proteins, although all membranes have lots of proteins, but here because I am talking about oxidative phosphorylation, the proteins involved in oxidative phosphorylation are present in this inner membrane. Now in the matrix, a cycle is going on that is more properly known as Krebs cycle after the name of its discoverer, Krebs, who discovered this cycle. And in the cytoplasm, a cycle is going on that is known as glycolysis. You know that carbohydrate metabolism has three parts. First part of carbohydrate metabolism is glycolysis. Second part is the Krebs cycle, and the third part is oxidative phosphorylation. In glycolysis, the glucose molecule C6H12O6 is converted into two molecules of pyruvic acid. In the Krebs cycle, a cyclic reaction occurs, and acetyl coenzyme A, which is a 2-carbon compound, combines with oxaloacetate to convert into a 6-carbon compound that is known as citrate. When a cyclic process occurs, this citrate again converts back into oxaloacetate. Right? Now during this Krebs cycle, the energy equivalents are produced, which are known as NADH plus H and FADH2. Similarly, during glycolysis also ATP is produced and another energy equivalent, that is NADH plus H, is produced. Here also ATP is produced. Now ATP is okay, ATP is the energy that is being produced over here and over here, which is very less. Here ATP is not produced, it is GTP. Right. Now ATP, GTP is okay, but what about this energy equivalence? One NADH molecule corresponds to about 2.5 ATP and one FADH corresponds to around 2 ATP, or you can say 1.8 ATP somewhere. This type of molecule is produced. Oxidative phosphorylation talks about how this 2 ATP and this 2.5 ATP are formed from the NADH molecule. See, I told you before, oxidative phosphorylation occurs in the inner membrane of the mitochondria. The inner membrane has large number of proteins, and the main proteins which are involved in the process of oxidative phosphorylation and in the generation of ATP are totally 5 proteins. And actually not proteins exactly, these are a complex of proteins named as complex 1, complex 2, complex 3, complex 4 and complex 5. One by one we will understand each and every complex, and then we will come to know the exact process of oxidative phosphorylation. Now, the first complex of oxidative phosphorylation, complex 1, is also known as NADH. Dehydrogenase complex. This complex, what it does, it takes up NADH plus H molecule, which I had written before. It takes up this NADH plus H molecule and converts this entire NADH into NAD plus molecule. This NAD plus molecule will now go back to the Krebs cycle for generation of another NADH plus H molecule. Now during the process, some electrons, 2 electrons will be produced. 2 electrons will be produced and 2 hydrogens will be produced. Right? This complex itself is a pump which pumps out these hydrogen ions out. Hydrogen will be pumped. This hydrogen, how it will be pumped? It will be first given to this FMN molecule. FMN is the primary acceptor of electrons, of primary acceptor of hydrogens in this first complex. Please note that this complex is a shoe shape, it is a boot shaped molecule, boot shaped protein, and this FMN gives the hydrogen ions further to gives the electrons. I was talking about the electrons. Electrons are taken up by FMN, which are further given to certain iron sulfur centers. I will write star over here. There are many iron sulfur centers over here, and from here it is given to a ubiquitin molecule. Now ubiquitin is actually, suppose this is the ubiquitin molecule. So this is a lipid soluble molecule. Right, so it is lipid soluble, therefore present between the two membranes, and this molecule is not transmembrane, it is not spanning both the membranes. This molecule can be transported from one place to another. So from here it takes up the electrons with it. It takes up these electrons with it, which I had written over here, and it takes up these electrons to complex number 3 and gives up the electrons to here. This is complex number 3 here. Now ubiquitin molecule finally coming over here, right? One more thing, from in glycolysis also NADH was there. So here also this NADH will come through the outer membrane to the inside, and here also this NADH plus H will convert into NAD plus 2 electrons plus hydrogen. So there is a lot of hydrogen. This hydrogen is left. This hydrogen is pumped out of this channel. Hydrogen is pumped. If I write it like this, changing the color, this hydrogen is pumped outside the complex 1 into this intermembrane space. So if I circle these, large number of hydrogens are present now in this intermembrane space. So actually due to this pump and due to these hydrogens also, the NADH which is coming from outside, the concentration of hydrogen increases in this area, means this is more acidic in comparison to this inner part, that is the mitochondrial matrix. Talking about the electrons have already gone to the ubiquitin, through the ubiquitin molecule to complex number 3, and complex 3 has a cytochrome C molecule attached with it. Complex 3 has a cytochrome C molecule attached with it, which the name of complex 3, first I'll talk about complex 2. Complex 1 is over. What happens? What about complex 2? See, if I draw it like this, this is complex 2. This is complex 2. See, this complex is also known as this is the ubiquitin molecule which gave the electrons to this complex 3 and came back into its position. Now talking about complex 2, this is known as succinate dehydrogenase complex. What this complex does, it converts the succinate molecule. If I write it over here, this complex converts succinate molecule to fumarate molecule, and in this process a FADH2 molecule is produced. This FADH2 molecule will further convert into FAD. FAD will go to the Krebs cycle back, and this FADH FADH2 molecule, FADH2 molecule will convert into FAD. FAD will go back to the Krebs cycle along with hydrogen ions. So these hydrogen ions will come to this side. This complex, there are two specialities about this complex. First thing about this complex is that its name is succinate dehydrogenase. This is the only enzyme. All enzymes are proteins and this protein itself is an enzyme. So this protein, this enzyme is present both in the oxidative phosphorylation and in the Krebs cycle also. This is the only protein present in both part of carbohydrate metabolism, first thing. Second thing, it is not a pump. It will not transport these hydrogen ions to the outside, because just I wrote FAD molecule, one FADH2 molecule converts into one FAD and gives around 1.5 ATP to 2 ATP, written like this somewhere. So this energy is not that much that hydrogen can be pumped out also, right? Now obviously some electrons will also be released in this process. So these electrons will also be transported to this, that is complex 3. Complex 3, which I had discussed before, is having complex 3, which I had discussed before, is having this cytochrome C attached to it, and it is known as cytochrome C oxidoreductase. It is known as cytochrome C oxidoreductase. Now as such, this complex has no functions. Overall function is this complex 3 has taken electrons from complex 1 and complex 2, given to cytochrome C molecule. Now again, this cytochrome C molecule is a movable molecule like ubiquinol. Suppose this is your cytochrome C molecule. It will take up all the electrons, with it, move from this position and go to this complex 4. Here it will come and attach. The cytochrome C will come and bind to this position and give all its electrons to this 4th complex, which is also known as cytochrome oxidase. Now oxidase, please focus on this word, oxidase. This is the complex where oxygen molecule is going to be. This is the position where the oxygen molecule is going to convert into, oxidizes into oxygen, or reduction of water molecule takes place. So when cytochrome C, after giving this, it will come back to its position over here. It will come back to this position, right? Now oxygen has converted into water molecule over here. So in this process also electrons and hydrogen ions will be produced. So overall, if you notice, there is a large flux of hydrogen ions on this side overall, and electrons are present. These are moving, or you can say still staying over here, but present in this intermembrane. These hydrogens are present in the intermembrane space. So by the end, when it comes to this part, complex 5, complex 5 is also known as ATP synthase. Complex 5 is again an enzyme which is a ATP synthase enzyme, and this complex is also known as F0F1 complex, right? This F0F1 complex on the inner side has these small molecules attached to this. These are ADP molecules, right? These ADP, now what happens when a large flux of hydrogen collects over here? This is not a pump. This complex is actually a channel. Through the channel, these hydrogen molecules start coming inside. This is a channel actually. So through this channel, these hydrogens will start coming inside. Over here, these channels will come and bind to ADP molecule and convert this ADP we will discuss later that total number of ATPs produced during when one molecule of glucose is completely utilized, then that is 38 ATP. So this is the place of production of 38 ATP, right? So this is known as ATP synthase. Sometimes question is asked in exams that at which complex oxygen is actually utilized. So it is complex 4 where oxygen is actually utilized. This is complex 3. Right. Now question is generally asked about DNP, dinitrophenol. Dinitrophenol is a uncoupler, DNP molecule, which is also known as dinitrophenol. See, if you notice this entire events which are occurring in the inner mitochondrial membrane. So what is happening in these all events? This oxygen utilization is coupled with the production of ATP. When oxygen converts into water molecule, then more oxygen is required, right? This ATP takes up electrons from this place. These electrons from here are taken by ADP. Hydrogen comes through this channel. So it takes electrons plus hydrogen, then converts into adenosine triphosphate, right? So the reaction is overall coupled. Dinitrophenol, what is it? It is an uncoupler. It will uncouple. It will uncouple these two reactions. So what will happen? Oxygen will keep, if we add DNP to this reaction mix, so oxygen will be keep on utilizing. It will keep on utilizing the oxygen, but it will not produce. The process will not produce ATP in any way or the other. Right. Now, one more question is asked: if we add a ATPase inhibitor. If we add ATPase inhibitor, then what will happen? If you add ATPase inhibitor, then ADP is converted into ATP. That is the last step. If you add a ATPase inhibitor, means this ATP is being produced but not being utilized. If ATP is not being utilized, there is going to be an excess of ATP out here. So no more ATP is required. In that case, oxygen consumption will be reduced. So oxygen consumption, if we write about oxygen consumption, if I write it here, oxygen consumed. So in presence of DNP, oxygen consumption will be increased. And in presence of ATPase inhibitor, oxygen consumption will be reduced, increased. Clear? So this is all about oxidative phosphorylation. If any parts are left, I will make one more video for better understanding. Thank you.